Good morning. First, I would like to thank the conference committee to provide this opportunity for me to report our study at this meeting. Today, I will talk about the metabolomic proteomic study in small cell lung cancer. The small cell lung cancer accounts for 15% of all new lung cancer cases and is sixth rank of cancer-related deaths in the United States. There's no significant change in the systematic treatment for the patient with small cell lung cancer and has a tendency to disseminate earlier. So more than 80% of patients diagnosed with extensive stage disease. So the early detection biomarker and the novel therapeutic target are urgent need in the small cell lung cancer. Our original question is, can we identify the novel kinase which drive the small cell lung cancer survival? So we applied the activity-based protein profile to study the small cell lung cancer. Here is the probe. The designing ATP binding probe can specify binding the ATP activity sites of the enzyme, include the kinase, metabolic enzymes. Then the binding can modify the conserved lysine in the ATP pocket. Here is the ABPP profile is the process. We make the lysate and use the ATP probe binding the activity enzyme. Then we use, after digestion, the modified the peptide can purified by the strip avidin beads. Then we use the LC-MS to purify the ATP binding protein. We characterized the ATP binding protein in the 18 small cell lung cancer and 18 non-small cell lung cancer. Here is the pie chart shows the ATP binding protein identified by ABPP in the 36 cell lines. You can most of them is the ATP binding protein, enzyme, and kinase. The heat map is generated based on the intensity of each independent intensity of the peptide. And you can see the upper right corner, upper left corner is the peptide with the high density in the small cell lung cancer, and the left right corner is the high density peptide in the small cell lung cancer. We total identified more than 16,000 highly expressed proteins in between the small cell lung cancer and the non-small cell lung cancer. We do the peptide enrichment analysis, and here lists the top 10 pathways in the 2 cell lines. The top 2 and 3, the significant pathways in the small cell lung cancer related to the glycolysis. And the top 10 the pathways in non-small cell lung cancer, mostly is related to the cell adhesion, but no metabolic pathways. So the metabolic pathways enrichment in the small cell lung cancer draw our attention. So we further annotation all the high expressed the proteins in the small cell lung cancer. The total the 42, the enrichment the enzymes is related to the several the metabolic pathways, include the 19 the enzyme related to the purine. The next is question is this enriched metabolic pathways in small cell lung cancer is due to their growth rate. So we examine the doubling time between the small cell lung cancer and the non-small cell lung cancer. The result indicated there are no significant difference between the small cell lung cancer in the doubling time. This suggests the metabolic pathway enrichment in small cell lung cancer is not due to the proliferation alone. In order to validate our finding in the ABPP in the cell lines, we collaborate with RTR International in North Carolina and characterize the metabolites profile in the 10 small cell lung cancer and the 10 non-small cell lung cancer. Sorry. This process, is some problem. The process is the cell line extractor identified by the UPLC, QTOF, and MS in the positive and negative ion model, use the reverse phase separation, and all the group differential metabolites identified by the two soft ways. Sorry, this is not shown very well. And this is the cell line metabolites result. The principal components analysis of the small cell lung cancer and non-small cell lung cancer metabolites profile indicated there are distinct separation of the two cell lines. You can see the non-small cell and non-small cell lung cancer have the different distinct metabolite profile. The analysis result indicated there are total 85 differential expressed metabolites between the two cell lines. Around the 31 metabolites high expressed in the small cell lung cancer. The 6 of them highlighted with the red dot is related to the purine metabolites, such as inosinic acids, adenosine monophosphate. So we draw the conclusion from our cell line is the metabolic enzymes identified by ABPP enrichment in the small cell lung cancer and involved in the several metabolic pathways. And the distinct metabolites profile in small cell lung cancer include the metabolites related to the purine biosynthesis. The cell line results suggest there are biological connection between the ATP binding protein and the metabolites in small cell lung cancer. The next question is this metabolic signature conserved in the small cell lung cancer human tissues. So we performed the ATP binding protein in the 7 small cell lung cancer, 7 non-small cell lung cancer, and 7 normal tissues. Here is the heat map. You still can see the differential peptide, the upper left corner and the lower right corner. Compared with the normal human tissues, there are 2,500 the proteins differential expression in the small cell lung cancer and non-small cell lung cancer. So the pathway analysis indicated the small cell lung cancer and the non-small cell lung cancer have the couple of the metabolic pathways enriched compared with the normal tissue. Also, you can see some enzyme is related to the two pathways are specifically identified in the small cell lung cancer. The one is the alanine aspartate glutamine pathways is the part of the purine metabolism. So this Venn diagram indicated the high expressed proteins identified in both in small cell lung cancer tissues and in small cell lung cancer cells. The overlap, there are 201 proteins, both are identified in both the small cell lung cancer tissue and the cells. Of them is the enzyme is related to the purine pathways. So we also performed the metabolomic studies in the same set of the human tissues. Here is the tissue metabolomic study result. The principal components analysis also indicated the distinct separation and clustering of the 3 groups, the normal cell, non-small cell lung cancer, and small cell lung cancer. In the small cell lung cancer, we identified the 47 high expressed metabolites compared with the normal tissues and non-small cell lung cancer tissues. We do the enriched analysis for these 47 metabolites. The result indicated a couple of the purine nuclear metabolites pathways is enriched in small cell lung cancer. This is the KEGG, the purine metabolism map. The rectangle represents the enzyme and the circle represents the metabolites. Highlight the original enzyme and the metabolites is identified in our experiment in both small cell lung cancer tissues and cells. So we draw the conclusion from our tissue studies is the tissue result is confirm our observation in the cell lines and the purine metabolism is enriched in small cell lung cancer. Here is our future direction. There are a couple of ways can move this project from the small cell lung cancer proteome and metabolomic studies. First, we generate the metabolic inhibitor library. It covers more than 80 the compound, targets 30 the primary metabolic enzymes. Also, we can use the custom siRNA library to knock down the interesting enzymes. So both screen can validation our interesting the enzyme related to the small cell lung cancer survival and growth. Also, we generate the small cell lung cancer resistant cell lines. Our collaborator have the PDX, the mice model. Both two can be used to further validation our interest in the enzymes. The third direction, the same high expressed the metabolites or enzyme can be considered to the early detection by mark. The fourth, but not the last, is related to the immunotherapy. The same highly expressed the metabolites maybe suppressed the T cell activation, so we can come in the strategy to reduce the metabolites and reverse the T cell function and promote the small cell lung cancer immunotherapy. Here is my boss, Eric Horrell. We have the good team in the Murphy Cancer Center. The Paul Stewart in Horrell Lab right now is working on the integration analysis of the ABPP and the metabolized data sets. And Anne Stephen right now guides the Paul to the integration analysis method development. We have the good collaboration RTR group. They provide the very quantitative metabolized profile the data sets. There are 2 grants support this study. I have the poster tonight, and you are welcome to stop by. Thanks for your time. I'm glad to answer your question. I'm sorry the one slide didn't work well. It's the human tissues and the human cell lines. It's not the plasma.
Other questions? So for early detection, it would be ideal to do something non-invasive like exhaled breath or something like that. Do you think there's any potential to do the same type of analyses using something less invasive than tissue?
It's a good question. We consider these ways. For the highlight expressed metabolites in the small cell lung cancer, maybe we can do the MS, like the RTR group can do the human, the blood, the plasma, the detection for the interesting metabolites. For the enzyme, we also can have the ELISA can do the validation in the cell culture, the supernatant. After that, if we can confirm that is high, really high in the small cell lung cancer, we can do the translation to the human plasma.